Hi, I'm Andie or Andrea Smidler, and I'm a postdoc at the Akbari lab at University of California, San Diego. I received my PhD from Harvard University in 2019, where I graduated from the labs of Flaminia catarrhea and Dr. George Church at Harvard Medical School. We develop genetically modified mosquitoes intended for release for population suppression. So how do we do that? So there's this concept called sterile insect technique, and the idea is that female mosquitoes only mate once. So if you can mate them with a sterile male mosquito, the female becomes completely sterilized for the rest of her life and she's unable to lay eggs. And an important point here is that only female mosquitoes bite and drink blood and spread disease. Male mosquitoes cannot. So that means if you can mass release sterile male mosquitoes, they find and copulate with the wild females, and the result is no eggs in the next generation. And so that means releasing these mass sterile male mosquitoes is an intervention, and it acts like a species-specific chemical-free insecticide. For this award, we were developing genetically engineered mosquitoes for release in Africa. And so these are CRISPR genetically engineered mosquitoes expressing CRISPR components, Cas9 and gRNAs. Specifically, this project uses a Cas9 mosquito line and a gRNA mosquito line, crosses them together, and the result is that all the daughters are killed. And so this is both a perfect genetic sexing strain, which is useful for mosquito releases, as well as its own standalone vector control technology. So in developing these transgenic, genetically modified mosquitoes, we are transgenically expressing guide RNAs. And so CRISPR-QC's role was to help us validate these guide RNAs and determine which ones were the best cutters. And so they were able to show us that the two guide RNAs that we were using in our transgenic line, one was a really great cutter and one was not a great cutter at all. And that correlated very strongly with our in vivo data as well. And that's why we love CRISPR-QC so much, because it really does appear like their in vitro assay closely mimics the in vivo function of these gRNAs. Working with CRISPR-QC was really simple and straightforward. They basically printed off or synthesized the gRNAs for us. And then all we had to do was provide a PCR template, so a purified PCR template. So we gave it to CRISPR-QC, and they combined them together on their chip assay and were able to tell us which gRNAs were really effective cutters. So before CRISPR-QC, we were really just limited to in silico gRNA analysis. We really just used whatever in silico optimization tools were available online to develop our gRNAs. But this was really suboptimal because we developed these really complicated transgenic mosquito lines that take a lot of time and energy to develop. And so to develop these lines over the course of 2 or 3 years and then discover that you have a bad gRNA being expressed is really kind of suboptimal. And so that's where CRISPR-QC is now part of our workflow for pretty much every project to make sure we get our transgenic mosquitoes right on the first try. And actually, one of the projects that we're getting off the ground right now, we are working with CRISPR-QC to validate our gRNAs, but this time we're a paying customer. Science is really uncertain, and I strive to eliminate as many variables as possible. By testing your guide RNA with CRISPR-QC in advance, you've eliminated one very critical variable.